Welcome to Notes number 10. Today we're going to be talking about DNA and RNA. We're going to be looking at the general structure of DNA. We're going to be identifying the purpose of DNA replication, and we're also going to be comparing and contrasting the structures and functions of DNA and RNA. If you remember, DNA is a nucleic acid. That's why it's called DNA. Nucleic acids are organic compounds that both store and transmit genetic information. Remember, if it's organic, it means that it contains the element carbon. Carbon is the most important element. There are two types of nucleic acids: DNA, which stands for deoxyribonucleic acid, and RNA, which stands for ribonucleic acid. DNA is responsible for storing genetic information, and RNA is responsible for transmitting genetic information. We're going to look at some differences later on, but quickly, DNA is double-stranded, D for double, and RNA is single-stranded. Nucleic acids are made up of nucleotides. So nucleotides are considered the monomers of a nucleic acid. Remember that mono means one. So just one of these is a nucleotide, and many of these are going to make up a nucleic acid. Nucleotides consist of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Remember that the 6 most essential elements of life are chnops, or New Hampshire cops. So the only one that is not in a nucleotide is sulfur. A nucleotide has 3 parts, and you can see them here. The first is a 5-carbon sugar, also known as a pentose sugar, because pent means 5. There's also a phosphate group attached, which contains phosphorus, and it has a nitrogen base. We're going to see that DNA has 4 types of nitrogenous bases, A, T, G, and C, which all stand for the bases. DNA has a very specific structure. The backbone of DNA, or the sides of the ladder as we sometimes call it, are made up of alternating deoxyribo and phosphate groups. So here you can see one nucleotide that we just looked at with the sugar, the phosphate, and the nitrogen base. So the phosphate and the sugar are going to be making up the rungs of the ladder, and the sides of the ladder, and the rungs or steps of the ladder are going to be our nitrogen bases. So you can see a nucleotide here, another nucleotide here, another nucleotide here, and etc. So just one is a nucleotide, and this whole thing is a nucleic acid. Now, if you saw in the middle, only certain nitrogenous bases match with each other, and that's because of the shapes of them. Purines complement or go with pyridamines, which means that adenine is only going to bind to thymine, and guanine is only going to bind to cytosine, because purines, which have 2 rings of carbon, and pyridamines, which only have 1, need to match up, because you don't want the steps of the ladder to be thicker or thinner. You want each of them to be the same, so you want one big molecule or purine to match with one pyridamine. So A always pairs with T, and G always pairs with C. I always remember it as at GC. When I was growing up, my town was called Glen Cove, so I just remembered at Glen Cove, and when I went to college, I went to Gordon College, so it worked as well. But you guys can think of it any way you like. So these are going to be matching up, A and T and G and C. They're going to be making what's a ladder, and when it twists together, this structure right here is called a double helix. So DNA is composed of 2 strands, and these are going to be held together with hydrogen bonds. When they twist together, again, this shape is called a double helix. DNA has to replicate or make another copy of itself all the time when we're making new cells and when we're making our sperm and egg cells to pass on to our offspring. So DNA replication is the chemical process that makes exact copies of DNA. What it's going to do is it's going to preserve gene sequences for many generations. What that basically means is that because we're making exact copies, we preserve our genetic code. DNA is replicated or duplicated most often before we divide a cell. So both in our body cells, which is through mitosis, and through our sperm and egg cells through the process of meiosis, which we'll learn about much more in detail later. DNA replication is really not that complicated of a process. You just need to know a few things. One, we already know that the two strands of DNA complement each other because of base pairings, A with T and G with C. The same is going to happen when we're making a new strand. The first step is that there are two strands of DNA are going to unzip, as you see here. And what happens when they unzip is now you have these lonely nucleotides waiting here. So what's going to happen is new nucleotides that are made in the cell are going to come and attach and start making a new ribbon or a new strand. So here we can see the new one being formed. And again, they have to bond G to C and A to T. So as this process continues, we're going to have two strands of DNA that are formed, one on this side and one on this side, as it unzips. Each new strand that was formed is going to keep its bond to that old strand. So each new DNA molecule is going to consist of one new strand and one old strand of DNA. Here's another representation of it. The old strand is represented in this purple-blue. At first, it unzips, leaving these unpaired things, nucleotides. And we have our base pairs, which are not labeled here, but they would have to match up appropriately. So as it unzips, new bases are going to match up. So if this was G on this side, then a C would also have to be bonded here. So you can see at the end, we're left with two DNA molecules because we replicated or duplicated it, and each one consists of an old strand and of a new strand. DNA replication is very accurate because of the complementary base pairing, also known as Chargaff's rule. Adenine will only go with thymine and guanine will only go with cytosine, again, because of the shapes of them. So very few mistakes can be made, although there are mutations and other things that can happen that can ruin the process of replication. Now moving on to RNA. RNA is a smaller nucleic acid that helps DNA to make proteins. Since it's smaller, it's only composed of one strand. The reason why we need RNA is that DNA is too big to leave the nucleus. Remember, the nucleus in a cell is where the DNA is found. So what RNA does is it basically copies DNA, and it is small enough to leave the nucleus and move to the ribosomes. Remember those tiny little dots that are making proteins. So the RNA is traveling through these ribosomes, and out comes a protein. Now, RNA is also a nucleotide, but it differs in a few ways. One, instead of having deoxyribose as its sugar, it has a ribose sugar. That just means that it has an oxygen right here, and DNA doesn't have an oxygen right here. It still has a phosphate group like DNA, and it still has nitrogenous bases, but it has one different nitrogenous base. There's still only 4 types of nitrogenous bases. They are adenine, uracil, guanine, and cytosine. So instead of having thymine, we replace it with uracil. So again, RNA uses uracil instead of thymine. So instead of having A, T, G, and C, we have A, U, G, and C. Still, A is going to bond with U, and G is going to bond with C. Like we said, RNA is single-stranded. It still has the phosphate sugar backbone with phosphate and sugars alternating, and it still has nucleic acids in the middle. But since there's not 2 strands, we don't really form a step or a rung of a ladder. So summing up the differences between DNA and RNA. The first difference is that DNA has the sugar deoxyribose. That's why it's called deoxyribonucleic acid. RNA has the sugar ribose, which is why it stands for ribonucleic acid. The other difference is the type of nitrogenous base. They have the same 3 bases except for thymine, T, and RNA has the base uracil, or U. Another difference is that DNA is double-stranded. Remember, it's in that double helix shape, while RNA is single-stranded. Not really usually, it's always single-stranded. So DNA has that double helix because it has 2 strands, and RNA does not have that double helix. In addition, as we pointed out in the beginning, DNA is what is storing the genetic information in the nucleus, and RNA is going to send that genetic information to the ribosomes so that it can make proteins for our body to function. And that's it for notes number 10.